To navigate the ocean of big data, artificial intelligence and machine learning have become indispensable analytical tools. These tools don't replace the doctor, but act as sophisticated co-pilots that analyze huge volumes of data, including medical images and patient records, to identify patterns and make predictions that support clinical decisions. Just think, according to estimates, the human brain is capable of storing 2.5 petabytes of data. Among these are all the pieces of information we need to move around and, in fact, all the various instructions for living. 2.5 petabytes is also the size of all the data produced by the Cancer Genome Atlas Consortium on 20,000 tumor samples. Given the sheer amount of data, we understand that a person alone wouldn't be able to make use of it, and this is where artificial intelligence can make a key contribution. If we provide a clean and specific data set, artificial intelligence can classify it with great accuracy, surpassing human speed and sometimes even human precision. The case of the Marlin classifier, based on neural networks and developed for acute leukemia, is an excellent example of success. Using DNA methylation profiles obtained through nanopore sequencing, Marlin is able to classify leukemia subtypes in real time, providing an accurate diagnosis in just a few hours instead of the days or weeks required by conventional methods. This demonstrates the enormous potential of artificial intelligence in delivering ultra-fast and targeted diagnoses, transforming data speed into immediate clinical value. EVO2 is an open-source generative artificial intelligence tool capable of predicting the shape and function of proteins encoded in DNA, and in the field of genomics, it represents a true milestone. Its fundamental purpose is to make the language of biology, the one we can write through combinations of A, C, G, and T, more accessible to all researchers. EVO2 was trained on a dataset covering nearly 9 trillion nucleotides compared to the 300 billion nucleotides used for EVO1. These 9 trillion nucleotides essentially include all known domains of life, from humans to plants to bacteria, even including extinct species. This immense amount of data allows EVO2 to analyze life in a universal way. To understand how EVO2 works, let's compare it to a natural language model like ChatGPT. If the human genome is a long book, 3 billion nucleotides and individual genes are the words that make it up. EVO2 tries to find syntactic patterns in this genetic code. If we want to design a new gene, we can give EVO2 the beginning of the sequence, and it will complete the gene in the same way a language model auto-completes a sentence. The usefulness of EVO2 also lies in its ability to compress the time needed for biological discovery, carrying out experiments that would take years in just a few minutes or hours. A tool like this could have an impact in many different fields. EVO2 could make improvements or write a gene in ways that have never happened in evolutionary history. By perhaps directing genetic mutations toward useful functions, it acts as an accelerator of evolution. In fact, after generating a promising sequence, EVO2 predicts how it will function in real life. And this sequence could then be synthesized and tested in the lab, even using genetic engineering. In the clinical field, EVO2 is exceptionally effective at predicting mutations that lead to pathogenicity and disease. EVO2 is skilled at distinguishing which mutations are random and harmless and which ones actually cause diseases like cancer. The hope is that this will soon have clinical significance. In the field of engineering, EVO2 can identify useful molecules and generate new genetic sequences to treat diseases or solve environmental challenges. Ultimately, EVO2 is like a biological time machine. Not only does it analyze the history of life through its vast evolutionary dataset, but it also gives us the tools to proactively design the biological future, steering evolution towards solutions that benefit human health and the environment. However, artificial intelligence in medicine is not a magic solution. It has cracks and inherent limitations that must be critically understood. An effective metaphor to explain the limits of deep learning is the idea of a machine that creates stereotypes. Take AlphaFold, for example, which was trained on millions of normal protein structures. So it has, in a sense, created within itself the stereotype of a healthy protein. When it receives a protein with a single deleterious point mutation, in fact, AlphaFold tends to predict a structure that's almost identical to the unmutated one. It fails to grasp the radical functional damage caused by the small change, because its knowledge base is dominated by the stereotype of the correct protein. This failure makes artificial intelligence still unreliable for predicting the structures of mutant proteins that are relevant, for example in cancer. When it comes to drug discovery, artificial intelligence has struggled with the de novo design of new molecules. In a study searching for molecules that could bind to the protein FIBP, a promising target for immunotherapy, the 10 most confident predictions made by artificial intelligence turned out to be disappointing. None of the molecules showed any significant binding in laboratory experiments. Studies claiming that artificial intelligence outperforms doctors in digital pathology are often flawed by unfair comparisons because they're based on a single pathology screenshot. On the other hand, a doctor builds their diagnosis on a complex system that includes multiple biopsies, immunohistochemistry, and the patient's entire clinical history. This still shows a gap between lab performance and clinical practice. The application of artificial intelligence in the medical field has exploded because it's a remarkable enabler of progress in clinical tasks like risk prediction and disease screening. The idealized promise of this technology is its objectivity and reproducibility, which should eliminate biases and clinical inequities, leading to personalized care for all patients. In reality, artificial intelligence models are often affected by bias, which makes them unfair towards certain groups of patients. In the context of medical artificial intelligence for clinical decision support, we define bias as any factor or prejudice that causes an artificial intelligence algorithm to produce unfair or distorted outputs and results. These biases are rooted in historical inequalities within healthcare systems, and if left unaddressed, a biased medical artificial intelligence model can lead to inaccurate clinical decisions and an increase in healthcare disparities. Bias can occur at four main stages: data and label collection, model development and evaluation, implementation, and research publication. The data collection phase is the first and most critical source of bias. Among the most classic examples of these biases is an unbalanced sample size, probably the most direct form of data bias, with a sample size insufficient for certain patient populations. Many datasets used to train clinical artificial intelligence tend to overrepresent non-Hispanic Caucasian patients. Not to mention the fact that many published clinical AI models use only data from the United States or China. Population biases are also widely debated in the GWAS studies, genome-wide association studies, where the poor representation of specific ethno-geographic groups or a predominance of males in the batches under examination can lead to incorrect results and conclusions. When an algorithm is trained on unbalanced data, it leads to worse performance and algorithmic underestimation for underrepresented groups. Other types of bias can be created by non-random missing clinical data, data that are not collected, such as social determinants of health, bias in data labels like diagnosis and mortality, which result from the subjective decisions of individual operators. To counteract the risks generated by bias, targeted solutions must be implemented throughout the entire pipeline. The most powerful solution is to collect large and diverse datasets that can represent and account for variations within and between patient groups. Where necessary, implement statistical debiasing methods, for example by using oversampling techniques like SMOTE and generating new synthetic samples for minority classes. Transparency and interpretability of the model are essential for developers and physicians as they allow for understanding how features drive predictions differently across various subgroups. In conclusion, artificial intelligence in medicine promises a future of optimized care. However, bias is a significant risk, not just a possibility, but a real threat that can alter clinical outcomes and worsen existing inequalities. Addressing these biases is crucial to ensure that all patients benefit equally from the future of medical artificial intelligence. The success of Marlin on one hand and the failure of AlphaFold on the other are not contradictions, even if they might seem so. Instead, they demonstrate the heterogeneous maturity of artificial intelligence, which excels at classification tasks with clean data but fails at complex generative or predictive tasks. The fundamental message is that artificial intelligence is a powerful and complementary tool, and never a substitute. It must be used with a critical eye, understanding its inherent limitations.